Okay. Well, thank you, Alan, and it's great to see everybody here, both in person and online. Thank you all for coming. We are about to start our 40th season of butterfly data collecting through the Butterfly Monitoring Network. And how did we get here so quickly? But um, I thought this would be a good opportunity to look back and see some of how over time the data have been used. So we'll see this graph again in a few minutes, but this is the number of surveys that have been submitted every year since 1987 when the monitoring network began. And you can see that the program has been growing and we've been getting more and more data over time. In fact, with the exception of the pandemic year of 2020, since 2019, over 1,000 surveys have been submitted to Pollard Base every year. We now have over 22,000 surveys in from Illinois in Pollard Base. So this is quite a hefty data set. And I think when we do the annual update in a little bit, we'll look in a little bit more detail at the data that we have received. The red arrow here is 2012, and that is the year that the very first peer-reviewed publication came out using Illinois Butterfly Monitoring Network data. So this is a graph from that particular paper. We're not going to go into the details on the paper yet. We're not going to go into the details of that paper in this talk. But it wasn't really about butterflies, their ecology, or their biology. It was about monitoring itself. And it was comparing monitoring in two large cities, Chicago and New York City. And different data collection protocols were used in both of these programs. We're, of course, using Pollard Transect here in Illinois, and in New York, they were using a count circle program. And it came out in the journal Conservation Biology. The graph here shows species accumulation curve. By adding more monitors, how many more species of butterflies do you see? And it shows that for both Illinois and New York, somewhere in the 50 volunteers, 50 monitors range is where you flatten the curve out.
Just want to fix it real quick. Onto the Zoom. So for this.
There we go. Oh, thank you very much. So this was the very first paper that came out from Illinois Butterfly Monitoring Network data. Since that time, we have had 28 publications that I have been able to uncover that have appeared in the peer review literature that have incorporated Illinois Butterfly Monitoring Network data. And so for the purposes of this talk, I would like to examine 3 of these papers in a bit more detail. And the 3 papers that I've chosen even come together to form a bit of a coherent story between themselves, even though it was not the same authorship teams on all of the papers. Now, I'd like to, here is the graph of the publications annually. Oops, hope that went. And you can see there was the first paper in 2012, and then a bit of a gap, and then papers have been coming out at a much more regular clip. And I think you can see a couple of things going on here. The first thing is it takes a while, quite a while, to get enough data to do any sort of meaningful analysis with. And part of the reason for that is that butterfly monitoring data tends to be very, very noisy. Insect populations tend to jump up and down annually because most species need to complete at least one generation annually. And so this means that the data just naturally tend to jump up and down a lot, producing a very noisy data set. It's because of that, it can be difficult to see the signal of something that you're detecting over the noise. And generally, what statisticians tell you to do, faced with such a data set, is to collect more data. And here in Illinois, we've done that by collecting data with more and more people every year. We've done it by collecting data for longer and longer periods of time. And as you will see with at least some of the papers that we're going to talk about, we have combined our data set with other data sets from outside of Illinois. The other thing that tends to happen and tends to stimulate more publication is once you start getting papers published and out in the literature that use the data set, more and more people become aware of the existence of the data set. More and more people ask to start using the data set for their own analyses. And there's a whole snowball effect where interest in this particular data set starts becoming much more self-generating. And so there have been, over time, we're accumulating more and more papers. Now, you might remember a year ago, the keynote address was about a paper that came out in Science, and that's a very, very high-profile journal. And so it'll be interesting to see what happens in the aftermath of that in terms of people using our data and publishing even more studies out of the Illinois data set. I wanted to just very briefly review the findings of last year. It was the subject of our keynote address last year. I want to just very briefly review it because it is related to a couple of the other papers that we're going to talk about. So this combined data set from 35 different programs and analyzed 342 species of butterflies and basically had very, wrong way. Depressing finding showing that between 2000 and 2020, there was a 22% decrease in total butterflies across North America, that many more species declined than increased, and the declines tended to be of larger magnitude than the increases were, and that the declines were scattered in all regions of the country. A few years earlier, there was a much narrower and more restricted study that showed something very similar. And this is a paper that came out in 2021, Butterfly Declines in Protected Areas of Illinois, assessing the influence of 2 decades of climate and landscape change. Now, this is a bit unusual relative to the other papers that we'll be looking at because this is the only one that uses Illinois butterfly monitoring data exclusively. It's not combining the IBMN data with any other data sets. And the time frame was for this study was 1999 to 2018. And you can see scattered around on the map there are 7 different sites, most of which are up in the Chicago area, that were used in this particular analysis. So why these 7 sites? Because there are a lot more than 7 sites that data are being collected on in Illinois. The time frame here, 1999 to 2018, was broken into 4 5-year time periods. And to be included in the analysis, a site needed to have produced data for a minimum of 3 visits in a minimum of 3 of the 5 years in each of the 4 time periods. And it was a little surprising at the time. There aren't a lot of sites that fit all of those criteria. And so the 7 sites that were most consistently monitored for those 20 years are the ones that were included in this particular analysis. And the sites that were chosen were Cuba Marsh, which is in southern Lake County, Somme Prairie Grove in northern Cook County, Bluff Spring Fen in western Cook County, northwestern Cook County, Fersen Creek Fen in eastern Kane County, Spears Woods in southwest Cook County, Hickory Creek Barrens in Will County, and Buffalo Trace down in Champaign County. And Buffalo Trace is different from the other 6 sites, not only in that it is further away, but it's in a much more agricultural matrix. The remaining sites are all in more of a suburban matrix, and this made some difference to the data. So something very similar was seen in this study to what was seen in the science study that we looked at last year. There was a decrease in both the abundance and richness of butterflies on these sites. Richness is a measure of species diversity. So the abundance over all of these sites decreased by 52%, and the species richness over all of these sites decreased by 27%. So this is more of the kind of bad news that we've been seeing and talking about for a while now, both about butterflies specifically and about insects in general. And this joined a growing body of studies really all over the world that are showing similar effects. The declines were observed in all functional groups that were analyzed on butterflies. And the functional groups that we are talking about here include things like, are they habitat generalists or habitat specialists? Are the caterpillars food plant generalists or food plant specialists? Are they univoltine, that is, they have a single generation each year, or multivoltine, in which they have 2 or more generations per year? And in all of these functional groups, declines were noted. One paradoxical finding from this study is that land use in the immediate area around within 2 kilometers of the study site was also analyzed as a variable in the statistical model. And increasing impervious surface had a positive effect on most groups. And this was a little surprising because increased impervious surface generally means development is happening. And in fact, in the cases of the 2 sites that experienced the most increase in impervious surface, which were Hickory Creek Barrens and Bluff Spring Fen, the changes are very definitely resulted to urbanization and development in the immediate areas around these preserves. And in general, one would expect that to have a negative effect on butterfly abundance. What is hypothesized to be being happened here is that the specific change that has been encountered over most of these sites that were studied in this paper has been a transition from agricultural to suburban rather than to dense urban. So there is some hypothesis here that this is a reflection of the negative effects of agriculture on butterfly population sizes. And sort of keep that thought in mind, because this is going to come into play in the next paper that we are going to look at. So the next paper that we are going to look at came out in 2024, and it is called Insecticides More Than Herbicides, Land Use and Climate Are Associated with Declines in Butterfly Species Richness and Abundance in the American Midwest. So this analysis used entirely butterfly monitoring networks that use the Pollard transect method of data collection. And it was the programs in Illinois, Ohio, Iowa, and Michigan. Illinois and Ohio produced data throughout the study period. The study period here is 1998 to 2014. Illinois, the program had been in existence since 1987. Ohio, the program had been in existence since 1996. Michigan and Iowa both started their programs in the 2006 to 2007 range, so they did not contribute data to the very beginning of this time frame. Data for this study are aggregated from the county level. So in Illinois, all of the sites in Cook County were now analyzed together. All of the sites in DuPage County were analyzed together, and so on. One of the things that they were, one of the things that the authors were using as a variable in their statistical modeling was pesticide data. And pesticide data was obtained by paid phone interviews. They actually hired two people, or two groups of people got paid here. They hired a professional marketing firm and they got paid. And then the marketing firm reached out to growers and interviewed them about their pesticide use. And the growers got paid for their time for the interviewing. The interviews were mostly with corn and soybean growers, and the model included six pesticide classes, 3 land cover types, and 6 weather variables. And the 6 pesticide types included 2 that were targeting weed control and 3 that were targeting, and 4, I should say, that were targeting insect control. And they fell into 5 categories. 2 of the categories were for weed control. One was for traditional weed control, as this existed for a number of decades prior to the study, where farmers would be going out and spraying their crops with herbicides to control for weed. The target spectrum was low. Typically, single weed classes were targeted by the insecticides. The frequency was high because it was done both before and after crop emergence, but the hypothesized impact to insects was low here. Beginning in the early 2000s, herbicide-resistant crops were introduced as a new technology. And then initially with glyphosate, other herbicides began being used in this way, but not until after this period of analysis here. And what would happen is that crops would be engineered to be resistant to Roundup, and then the fields would be treated with a broad spectrum herbicide, Roundup, that the corn or soybeans would be resistant to, and so it would kill the weeds and leave the crops alone. The frequency of herbicide application was low here. It was mainly a post-emergent treatment. The hypothesized impact here was also low, with the exceptions for certain specialized insects, in particular the monarchs. This is the source of the milkweed limitation hypothesis for monarchs, where milkweed in agricultural fields was declining as a resource for the caterpillars. For insect control, there were 3 classes of situations that were looked at. The first was sort of your typical insect spraying, has been done for a long time with 2 different insecticides, pyrethroids and organophosphates. One of the things that is of concern with insects, because they can directly kill butterflies, concern for this analysis, is drift and spillover, which can kill things that are outside of the agricultural field being treated. There's a high risk of spatial spillover here, mostly through pesticide drift. The between season frequency is lowest in this because it's a reactive method. This would be used in response to ongoing outbreaks of insects. The persistence of these insecticides is low, and the hypothesized impact is fairly low because most of the impact on butterflies is going to be simply from drift off-site of the insecticide. Then there is another genetically modified crop, and this is corn that is engineered with a naturally occurring insecticide from bacteria. And this is expected to have low spatial spillover, and the insecticide remain very tightly associated with the agricultural field. This is an every year program because it's prophylactic. This is done not in response to insect outbreaks, but to prevent them. The persistence of this is going to be low in the environment, and the hypothesized impact is going to be low. There was some concern about monarchs when this technology first was introduced, but further study of this showed that it wasn't really affecting monarchs at all. And the hypothesized impact to non-target butterfly species was low. And finally, neonicotinoids. And neonicotinoids are a relatively new class of insecticides. They're characterized by being having a very, very high level of toxicity, such that very low quantities of them can produce very high levels of toxicity. Spatial spillover here is moderate to high, although it's a seed treatment. It can wash off of the seeds into the environment. The toxin is then translocated into the germinating plants and into the growing plants throughout the growing season. The between season frequency is high because this is a prophylactic treatment that is used every year. The persistence of the effects of neonicotinoids tend to be very high because they will persist throughout the entire growing season, unlike some other insecticides that are used. And the hypothesized impact is potentially high. So what did they find? Well, the three columns of charts here that you see are on the left, the effect of on species richness. The middle is the effect on total abundance, and the right is the effect specifically on monarchs. And so what they're using is the frequency of application of each of these treatments in the model, and then the number of butterflies that the monitors were observing were also included in the model. And the dotted line down the middle of each squares indicates a zero change. It would mean that this would have no effect. And what they found was that for most of these pesticides, the effects were not statistically significant. There was a small but statistically significant decrease in species observed with the traditional non-glyphosate herbicides being used. A paradoxical increase in species richness with with the new Roundup Ready corn and glyphosate being used, probably because fewer other pesticides were being used in conjunction with this. Neonicotinoids jumps out as the one that has the biggest negative effect on butterflies. The effect is statistically significant for species richness and for the monarch. The other things, factors that were looked at in these three cases were land cover in the area immediately surrounding the study sites, and then weather conditions each year of the study. GDD is a measure of temperature, and it's going to be low in it was looked at in early, mid, and late years, and a low GDD means it's cold in those times of the year. A high GDD means that it's warm. And GDD was positively associated with species richness for most species, but this effect was not statistically significant. It was negative for total abundance, and this effect, however, was only statistically significant for low temperatures early in the season. which coincidentally is something that we have anecdotally noticed in the butterfly monitoring network data here in Illinois. Temperature mid-season, low temperatures mid-season had most of an effect on monarchs. You wouldn't really expect the early season temperatures to have much of an effect on monarchs because they're not here yet early in the year. Precipitation tended to have a negative effect on butterfly abundance. And this is something else that we have noticed in the past, that very wet years can be bad years for butterflies. This puts some actual rigor to that observation. And when you do a combined model that looks at the percent control effect on population size with herbicides, insecticides, and combination of the two, you see that the insecticides are really having most of the effect. And most of that effect from insecticides is coming from neonicotinoids. And so the main conclusion of the paper is that neonicotinoids seem to be what's driving a lot of the butterfly declines that have been being observed in some of the other studies that have been reported. And this does sort of go along with the Illinois study that showed that decreasing in agriculture in the area immediately surrounding the study sites seemed to improve butterfly abundance. So those two observations seem consistent with each other. Final study I want to talk about came out also in 2024, and it's called Phenological Consistency and Management Interventions Predict Population Trends in At-Risk Butterflies in the United States. This used 3 main butterfly sets and then a bunch of much smaller sets. The big sets that it used were the Illinois and Ohio IBMN numbers, and a data set from California that was assembled by Art Shapiro, a faculty member at UC Davis, and a big name in butterfly ecological studies. And the Shapiro data set was collected with a very different data. He had about a dozen specific study sites identified across California. They were chosen very deliberately to be at different elevations at a similar latitude. And for a single person collected, this is not a community science program. Art Shapiro was pretty much collecting this data by himself and just going out a lot of times to all of these sites each summer. It's an amazing effort from a single individual to get a data set as large as he was able to get. The analysis emphasized at-risk species. At-risk in this case means anything that is listed as endangered or threatened at the either the federal or the state level, and also anything that is listed by listed as a species of special concern. And there are several variations on that terminology that get used, but they all mean more or less the same thing, which is there's no legal protection for this species, but we know it's in trouble, sorts of things. For this study, there needed to be at least 7 years in the 10-year time frame with 3 or more days that had positive counts for the species involved. And so there were 114 species and site combinations that had enough data to be included in this analysis. This included 31 at-risk species and 91 unique sites. So that's the take on the data that could be pulled out that was useful for this particular analysis. I wanted to say something about phenological constancy. I'm not going to go into the details on the analysis as it was done for that, but phenology is an observable biological phenomenon that repeats on an annual cycle. So the date that the swallows returned to San Juan Capistrano is phenology. The first date that you see a mourning cloak butterfly in spring is phenology. The peak flight time of Baltimore checker spots. All of those are elements of phenology. And what you might expect in a warming climate is things like the first appearance of a species, adults of a species of butterflies might often advance to earlier times in the year to go along with the fact that the beginning of spring seems to be advancing to earlier times in the year. And so species that do move their phenology in response to a warming climate are not showing phenological consistency. Species are showing more phenological constancy, I should say. Species are showing more phenological constancy if things like first flight, peak flight, that sort of thing don't move very much, even though the onset of spring seems to be advancing. And what was found relative to phenological constancy is that species are doing better to the degree that they are managing to maintain phenological constancy in the face of a changing climate. Management was an interesting thing to try to analyze here. And so remember, we're using sites mostly in California, Illinois, and Ohio, but a few other sites scattered around the country. And for each site, managers and volunteers were interviewed to identify years of mowing, haying, prescribed fire, herbicide application, grazing, planting of butterfly host plants or nectar plants, or other activities. And most prairie planting involves some combination of host and nectar plants. It was very difficult to assemble this part of the data set for analysis. And the reason is, if you think about it, in Illinois alone, there are dozens of landowners that we are working with on IBMN sites. A lot of the sites are forest preserve sites, but there are a lot of other different land managers at all. There are land foundations, there are state parks, there's IDNR sites, there's just a whole range of different land managers. Often all of them use different protocols for keeping data about what they've done. And often there has been, in the period of time that was being analyzed here, personnel turnover. And the person who was keeping tabs on data about management activities at the end of the time frame might not have been keeping their records in quite the same way as the person doing it at the beginning of the time frame. So all of these things contributed to a challenge in assembling management data. And we were limited in the quantity of management data that we were able to assemble. So initially, some of these things in that first bullet point, the mowing, haying, prescribed fire, and so on, the hope had been to be able to analyze each of these management techniques and see what they were doing. And we simply were not able to get enough consistent data to be able to do that. So what we ended up doing in the end was lumping them. And we simply said, did any management at all of any of these sorts happen in any given year of the time frame that we were now analyzing? And so what we were actually using for that part of the data set was what fraction of the 10-year time frame, what fraction of the years actually had some sort of management applied to them. And Illinois tends to be very heavily managed. So a lot of sites, it was every single year. But across the data set, there was a range of amounts of management that had been applied. And this is what you see when you do the analysis. And as you see, the two biggest frequencies, the two most frequently reported frequencies of management intervention are every year, which would be 1, and not at all, which would be 0. But there's a lot of points in between too, and it was enough to actually do some analysis. And the result was actually really good news. The result is that management works. And I'll read 3 quotes from this particular paper. Our analysis indicate that the management approaches selected at these sites were broadly helping improve population trends. A key finding was that active habitat management was strongly associated with increasing populations. And finally, we do not dispute the large-scale influence of global climate change, but our findings strongly suggest that local-scale management can mediate the effects of climate change and other threats of global change. And I wanted this to be the last paper that I really delved into because it is the least depressing of all of them, and actually offers a little bit of hope in the face of a lot of bad news about butterfly populations. So, do the findings of these study begin to create between them a consistent story? These are different studies with different authorship teams that used often very different statistical analytical techniques, but are they reaching conclusions that are wildly at odds with each other? And the answer to that seems to be no. There are some things that appear to be a bit inconsistent, but in the main, they are showing a consistent story. These, and actually a bunch of other studies, document ongoing declines in butterfly abundance. That pesticides, in particular neonicotinoids, seem to be playing an important role in the declines that are being observed. And finally, habitat management can reduce the declines that are being observed. I know a bunch of the people who are attending this do habitat management, so feel encouraged that the work that you're doing is important in a lot of ways, but one of the ways it seems to be important is for butterflies. There are also some observations regarding the data set that I'd like to make. And this actually also extends beyond the studies that I've actually talked about today. There are, as I said, 28 peer-reviewed studies that have been published that use IBMN data. So it's a big data set. We've known that for a while, and it gets bigger every year. And I think Alan is going to have a few words to say about the data size this year. But it's a large data set. One of the things that I think the specific studies are a good illustration of is the fact that even from a large data set, the amount of data that's actually used for a particular study often gets whittled way down from things like the time frame. Were enough surveys done on the sites that exist in that time frame to do analysis of. It also is one of the reasons that analysis can be very, very difficult to do on some of the rare species using this approach, because as those of you who are monitoring on sites that have rare species know, you often don't see very many of them at all. And so there aren't enough data points to really do much with for some of them. However, important features of the biology and ecology, as well as long-term population trends in butterflies, have been detected using the IBMN dataset, and this seems to be only increasing over time. One of the things that I was most surprised about, that I would not have predicted this 40 years ago when I started working with the Illinois Butterfly Monitoring Network, is that it would not tend to be used as a standalone data set, but that it would be combined with data that was being collected elsewhere in the country. And that with data, you can, the bigger your set is, the more statistical analysis you do, the more robust your statistics can become. And people take advantage of that and are combining data from like the upper Midwest or across the country. In short, the more data you have, the more you can do with it. And I think that that brings us to a close here, and I would be happy to take any questions. If you are online, I can't hear you. Yes. So if you have questions and you're online, please unmute yourself. Yes, you can also ask questions in the chat. Are there any questions in the chat? Okay, thanks, Alan. So does anybody have any questions? Yeah, the question is the drift insecticide, is that through the wind and the rain? And the answer is yes, in particular wind. And when you get licensed to be a pesticide applicator, and they teach you about how to do it, wind and drift management are big parts of the training. I think they're talking about that one. Yeah. Is that the question just to show it again? Okay. I don't remember whether Medewan specifically was included in the study or not, because what we did was we lumped everything by county. I know there were a bunch of Will and Grundy County sites that were included, but I don't know offhand specifically which ones were. All right. Well, thank you very much. I think Alan, oh wait, I've got a bunch of stuff in the chat here. Are there? Yep. Come on. Here we go. Okay. Yeah. Come on. Yeah, yeah. We'll get to all of these. There's some good questions here. Okay, there's a comment here. There's a bill in Illinois legislature to limit or restrict neonicotinoid use. SB 3200 in the Illinois Senate has been assigned to the Agricultural Committee. This bill, sponsored by Senator Julie Morrison, will be discussed in committee this week. Haven't heard yet about its status. That's good news. I know people at the legislative level, there are people who are very concerned about this. There is sort of the expected industry pushback, and it's going to be interesting to see how it plays out. Has there been any response from land managers to the 2025 paper? I assume the 2025 paper is a science paper. There's been more response from land managers to the paper that says that management works, not surprisingly. But land managers are aware of not only the 2025 paper, but a lot of the other literature saying that there are butterfly and insect declines that have been detected. Yeah, SB 3200. Are there studies looking at the effect of mosquito yard spraying on butterfly abundance? I know there have been studies out of Florida about effects of mosquito control, none up here yet. And um, the ones in Florida say what you think they're going to say. It's something I'm certainly very concerned about. Last year, one of the companies actually put one of their advertising signs on my yard. I've got a corner lot, so I'm high visibility. It lasted about 15 minutes before ending up in my trash. Oh, we got one new. Hang on, let me scroll here. Can you elaborate on habitat land or land management best practices, for example? I would like to have been able to elaborate in more detail on best practices based on the paper. I think it would be I think one of the things that that paper really showed is that there would be a real need for a more standardized way for landowners to record um, their management activities. I think that would, um, and of course, not only a standardized way of recording it, but some sort of a repository for the data, sort of like a Pollard base for land management activities. That would be great. Just from the literature, and this is not specifically from any of these studies, but just from what I have read in the literature, um, best management practices include things like burn, but don't burn too much. There seems to be a consensus that has been reached in the literature that in many cases, a 3-year rotation is good. A lot of the literature really recommends mixing it up and combining different management techniques on the same site, leaving something we don't talk about much here in Illinois is leaving permanently unmanaged refugia within preserves for insects. And that's something that I think could really do with a little bit more study. But I think we're still learning about what the best practices relative to insects really are. And that seems to be all of. I'm assuming you kept using a Pollard base easier to do that at a point. What is that? Actually, Pollard survey is harder to do. It's a more involved protocol. What the basic comparison conclusion that was reached out of that paper was that with count circle, you can accumulate species faster because in general, in some place like New York, within your circle, if there's someplace somebody has been seeing something rare, you can specifically go there to try to find it. And that's not a flexibility that Pollard offers you. On the other hand, Pollard tends to more rapidly accumulate data that is more powerful and more amenable to statistical analysis because it is more rigorously quantitative. So there are strengths and drawbacks to each of the two methods. And I think that's, these are only two of many methods that are used for measuring butterfly population size. And they all have their strengths and weaknesses, and they all have situations where they are the preferred method for applying.
where they gather data from Georgia, IBMN, 26 yards, and they're finding high levels of erythroid insecticide in all the data.
Yeah. Yeah, and this is from when? Yeah. Yeah. So what was just said here is the Xerxes Society is beginning to do data collection and study on this, and it sounds like if I'm understanding correctly, they're just sort of at the beginning of that process. And my prediction it's going to be a while before they're able to get enough data to see what the trend from it actually is. Yeah. But at least they've started. Oh, we've got more questions here. May we share your presentation with receptive land managers in Southern Illinois? Absolutely. And
All right, we are back. And Doug is up for the first part of our season update. Just want to get to that.
okay, so this is something that we typically do every year is give a little recap of the data from the previous year. 2025 was a very good year for butterfly monitoring. We received data from 1,172 surveys through the Illinois Butterfly Monitoring Network. This represents over 1,100 hours. We detected 87 species this year, which is actually very much kind of in the range that we have been detecting the last few years. We counted over 40,000 butterflies this year and saw 38.1 butterflies per hour. Now, one of the things that's interesting is if you look at the cumulative data, we have over 22,500 surveys from Illinois in Pollard Base, which is outstanding. We have 3.1 years of monitoring effort. It means if one person were to collect all of the data in the Illinois Butterfly Monitoring Network, they would have to start and do nothing else, including eating and sleeping, for 3.1 years and just collect butterfly data to get as much data as we've got. We've had over time 120 species, and Alan is going to say more about this in a minute. 2025 was a very significant year because our millionth butterfly got counted in 2025. That's quite the milestone. 37.5 butterflies per hour. As you can see, activity remains strong. 2025 was the year in which the largest number of surveys to the network was deposited in Pollard Base, so that's exciting. We did not spend quite as much time in the field to get to that number of surveys as we have gotten in a couple of the last couple of years. One of the things I remember very clearly about 2025 as a survey year is that it got off to a really low start in the early parts of the year. Most people were seeing very few butterflies, and that may be contributing to the lowered times. It just takes more time to complete a survey if you're having to stop and record a lot of butterflies. Species detected. This graph I always think is really cool because it really shows you how many people you need in the network in order to pretty much get all of the species that there are out there to see. In the first few years of the network, we only had like 15 or 20 people collecting data. It seems to be about 45 people. Once you get to that level, adding more people is not going to get you to see more species. It'll get you to see more butterflies, just not more species. So that's good. It means we're seeing, a good representation of what's out there. And here are the total butterfly observations that have been made in the network. And you can see this is not corrected, Alan, am I correct? Not corrected for effort. So this is just the total number of butterflies that are counted. So that big increase at the beginning that you see there is really just the growth of the network during that period of time. And I will hand it off to Alan here.
All right, so with the addition of the millionth butterfly observation into our data set, I thought it would be fun if we could try to narrow down what butterfly was it? What was the lucky butterfly? I don't have the exact answer for you, but I'll tell you how close we could get. We were able to identify the survey that submitted the millionth observation. So if we rank all the surveys by start time, we can look at the running total of total observations across the network. It was this one. So on July 13th last year at Miami Woods, thank you, Michaelan, you counted the millionth butterfly. Now, which one was it? One of the butterflies on this survey. We're not able to tell what order the butterflies were seen. So whichever one it was the 25th butterfly that was observed on this survey. 24 of them being cabbage whites. I think that's a good candidate, but honestly, the monarch is a good candidate as well. Yeah. So here you go. One of these. All right. So now we're looking at that total butterfly observation charts, and this time it is corrected for search effort. So if you divide those total observations by the total amount of time searched, so the number of hours, you get this figure. And this is just in Excel doing linear regression here. This is not a formal analysis. We have not narrowed down that data set to only include the sites that fit strict criteria. This is rough and dirty, but we are able to see that decline at a rate of negative 0.238. That's about 23%, annually, and that's similar to what Doug had shared in previous analyses. So kind of a bummer, but we would not be able to track this if it weren't for everyone here in the network. I just want to point out this dot right here at the end, that was last year. It was a little bit up from previous years. If you go back 2 years ago, it was pretty depressing that year, wasn't it? All right. And then I also went ahead and pulled what were the top 10 observed butterflies, just so you can get a feel for the numbers that we're counting. Of course, cabbage whites are the big winner here, at over 14,000 counted. And then we're going down 10,000 less to the next step. Common and orange sulfur were pretty high. I'm not going to read the entire list, but I just want to point out monarch came in at number 4. They were in our top 10 last year, but they were much lower on the list. It was a pretty great year for monarchs. I am eagerly awaiting the news of how the populations fared in Mexico. I haven't seen the formal numbers yet. Yeah, I've been seeing some information about their migration back northward, but hopefully very soon we'll get some of that data on how much area they were occupying in their overwintering grounds in Mexico. I hope it was up. As long as nothing strange happened, it should be up. Okay. And with that, we have our standard updates. Real quick, let's see if we got a question. Oh, we got a question. Was the gem satyr in the opening slide found on someone's route? And Doug can answer this. Yes.
Doug can answer this because it's my photo. That photo was taken when I was down in Southern Illinois. Pat Dunbar was with me, and we were at Heron Pond successfully looking for Creole pearly eyes, and I happened to take a good picture of a gem satyr, but that was not taken on a route.
But I will say, for our trainings, we have, the top 25 butterflies that we teach new volunteers to really study when getting started. For Southern Illinois, we have to have a top 30. And after our first couple of years there, we learned, okay, we have to add the gem satyr to that list. So that is, a pretty common butterfly on those routes, or some of those routes. Okay. So without further ado, I have some announcements to make about service awards. So we like to recognize volunteers who have reached important 5-year milestones of service. Milestones, which are 5 years of service as a volunteer monitor. So we have monitors who have been with us for more than 35 years. Now, this is our 40th season, so we don't have a 40th pin to award yet, but spoilers, we will next year. They're in the room with us. It's Doug Taron. So an update on service pins. Last year, I sent out a survey for folks who would like to receive service pins who had never been able to make it up to the Peggy Notebaert Nature Museum at our annual meeting to receive their pin. They were mailed out. Finally, we had a chance where I could mail out some service pins. For everybody who had not received some pins or you think you should, I will be mailing that form out again along with the recording of this meeting next week as a follow-up. So you can get a chance to fill that out as well. But let's go ahead and announce who should be receiving pins from their service last season. We have quite a big cohort that is reaching that 5-year milestone. So I'm going to read off the names here because you deserve it. So you have Amal Hassan, Amy Hank, Bob Coons, Celeste. I'm so sorry. I'm going to butcher your last name, so I'm going to continue. Chris Slattery, Danielle Green, Gary Janke, Jessica Johnson, Maria Nietos, Mary Ann Maglia, Mary Zander, Marianne Shabelsky, Rob Coolney, Susie Jizik, and William Sowers. Thank you so much, everybody, for reaching 5 years. Okay. Thank you. We have some 10-year pins to award, and those go to Gary Morrissey and Sue Morrissey. So thank you so much for 10 years of service. Moving on, we don't have any 15 years, but we do have a couple 20-year pins to award. Those go to Jeff Chapman and Penny Vanderheiden. We have a number of 25-year pins, believe it or not. Good cohort. Nanette Kalsher, Phyllis Schulte, Sally Wyclaw, and Susan Page. 25 years is really incredible. And there's one more. 35 years, we have Andy Kraut. So, Doug, some folks are coming out for you. All right, Doug, do you remember where Andy is off the top of your head? All right, Andy is at Spears Woods. All right, and I just want to highlight, I looked through and made this little chart so that we could kind of gauge the experience of the monitoring program as a whole. And, we have a large cohort of newer volunteers who have 1 to 5 years of experience. But I just thought it was really worth highlighting that over slightly more than half of our network has been with us for more than 5 years. So really, you guys hold such a wealth of knowledge, especially at your specific sites, because there really aren't that many people who are going to the same site systematically multiple times a year and really paying attention to what's there, recording the data, getting to know these specific taxa. Like, you guys are developing expertise that lives with you, and thank you so much for sharing all this information back with us. Okay, and oh, I just spoiled it, but we have an announcement. I'll bring Doug back up here because right after this, he has another quick topic to cover. Sulfurs.
Yeah, so we are going a little further afield this year to the Peoria area. And there are several sites right near each other. What they all have in common is that they're sand habitats. And so there are some species that are found there that we might not get to see up here. There's more southern stuff. It's also a little bit more central, so it's not quite as far if people want to come up from southern or western Illinois up there. So we are targeting meeting at Sandridge State Forest. There is also scrub oak sand savanna down there, and some really good stuff has been seen there. At this time of year, you see things like Olympia marbles down there. There have been regal fritillaries reported down there. So there's potential to see some really cool stuff. Oh, yeah. Rain date is the 12th. So we learned our lesson at Iroquois Prairie a few years back.
Here, Doug, I'm just gonna switch it to your skippers, but I saw we had something came through in the chat, so I wanted to check that out. Oh, they do remember. And more burning discourse. Okay. PowerPoint. There you go.
Thank you. Yeah. Oh, yeah. Okay. Let's see. Close the chat and switch to slideshow. Nope. Good. So this is a brief talk that I want to give. This is an identification talk called Some Tips for Sulfurs. And this butterfly in this picture is not a sulfur, but it is a tip. I'm not doing a travelogue this year, but I wanted to get to show at least one photo from my recent trip to South Africa, and the purple tips were just magnificent butterflies that I've always wanted to see and never gotten to see before. So we're actually going to talk a little bit about the sulfur butterflies in Illinois. And I wanted to do a quick recap of them because I've been noticing some, as the data come in, I feel like a bunch of people are struggling with these a little bit. In particular, up here in the far northern part of the state, I'm seeing lots of reports of cloudless sulfurs. And I am questioning some of these identifications. People are reporting cloudless sulfurs and not reporting the much more common and orange sulfurs up here. So I thought what I would do is let's take a look at all of the sulfurs. There are, I believe, 7 species here in Illinois. And just really quickly go over to maybe get people ready for the season here. So most of the sulfurs are yellow or white or orangish. And because of that, in part, this is a group where it's really important to use more than just color in making your identification. I think that this can contribute to sometimes more reporting of things as cloudless sulfurs that are necessary, because it's the one that is just yellow. It's the all yellow sulfur. And it's tempting to say, oh, look, a yellow butterfly is flying by. It must be a cloudless sulfur. Size really is an important addition to help you identify sulfurs. Size easily distinguishes 4 of the species of sulfurs, cloudless sulfurs being one of those 4. So all sulfurs in Illinois are bright yellow or orange, except for some species also have white females. So let's dig into them a little bit more here. And you'll notice that I'm going to be emphasizing here in the first few size more than the details of wing markings, because you can really, with sulfurs, that's probably, in addition to the fact that they're yellow or orange, it's the first thing you should be considering about them. So for example, the dwarf sulfur is tiny. This is a bright yellow butterfly that's the size of a blue. There is nothing in Illinois other than the dwarf sulfur that is going to have that combination of characteristics. Something that is the size of a blue, but the color of yellow that the sulfur butterflies have. And in fact, that combination of size and color is unique in North America. This is not a super common species in Northern Illinois. It's a migrant from further south. And when you see it up here, you're most likely to see it in migration corridors, which means on the lakefront, I've seen it, and also on river corridors. I've seen a whole bunch of them puddling on the banks of the Illinois River in Ottawa before. And that's very characteristic of one of the types of places you can see them. But they're dispersers. Individuals can show up away from these migratory corridors, but those are the places you're most likely to see them. And we're going to gradually work our way up the size category here. Another small sulfur is the little yellow, but this is bigger than the dwarf sulfur. This is the size of a pearl crescent. So it's bright yellow, and it's noticeably smaller than most of the sulfurs that you will see here in Illinois, but it's definitely bigger than a blue. It's definitely bigger than a dwarf sulfur. And they are this sort of medium yellow color. A lot of them out near the edge of the wing. Some of the females are pale. Ellen, you may remember when we took this picture. This picture is from Braidwood, and there was the host plant for little yellows is partridge pea. And we were in a field that was very, very dense with partridge pea. And we were seeing some females that looked white, especially, we were seeing large numbers of these, especially in comparison with the brighter yellow ones that were being seen. When they landed and you could take a picture of them, what you would see is that they actually are yellow. It's just a much paler yellow than most of them. And this is a common feature in sulfurs. A certain percentage of the females in many species will either be white or a much paler shade of yellow. But again, the size here, you're looking for something that's about the size of a pearl crescent. Sleepy orange. I have seen one of these exactly in Northern Illinois ever. It was right over there. Well, it was probably using the lake as a migration corridor, and it got into the ravine here at the Nature Museum, and we got to see it taken. I only had my phone camera, so I didn't get a very good picture of it. However, this is a common butterfly in Southern Illinois, and this is going to be noticeably smaller than a cabbage white, but it's going to be bright orange, especially as it's flying. As you can see in this picture of the perched individual, it's kind of a brownish yellow orangey color, but the upper surface is bright orange, and that's what you really see when it flies by. In Illinois, this is the only orange sulfur in this size range. It's the only orange sulfur that's going to be visibly smaller than a cabbage white. Now, I'm going to say more about the really common medium-sized sulfurs in a minute. I'm going to go through the other ones first, but there is one other medium-sized sulfur that's about the size of a cabbage white, and that is the southern dogface. This is a rare migrant. The last time I saw one in Illinois was in 2016. So, they'll go many, many years without seeing them, and then they'll have a population explosion and they will show up. And I've said here, it's uncommon until it isn't, because they use lead plant and purple prairie clover as host plants. And so in 2016, what happened is a few of them got up here early enough in the season that they were able to breed here that year and had a couple of generations. And at Bluff Spring Fen, by the end of summer, this was the most common sulfur at Bluff Spring Fen, which is wild. That never happens. So under unusual circumstances, you might see it, but most people in most years are not going to see it. One of the things to notice about one of the things to notice about the southern dogface is that the tip of the forewing is shaped very differently from other sulfurs. In most sulfurs, I'll go back one here, you can see it's got a rounded forewing tip. The southern dogface comes to a definite and pronounced point. Yes, it gets its name from the pattern of the dog's head that the dark edges form on the upper side of the wings. The thing is, you really don't get to see that. This species, and indeed most of the sulfurs, do not land with their wings open. And you can't see the dog's head very well as the butterfly is flying past. So in practice in the field, it's the wing shape that I have typically used when I look at these. I get to see them a lot more often because I go to Arizona a lot, and they're common here, but they're not common here. And finally, the cloudless sulfur. And this is big. This is going to be visibly bigger than a cabbage white butterfly. No other sulfur in Illinois is this large. It is bright yellow. Alan has called it the flying highlighter, and I like that. It will often not land. It will often just be sort of flying through an area, often at about head high. And the more common sulfurs, when you see them, they tend to be lower, and they tend to be very skittish butterflies. They'll land in nectar and move a little bit and land in nectar. Occasionally, the cloudless sulfur will do the same thing, but most of the ones that I have seen, the one in this picture being a notable exception, are either flying and just sort of going through an area, and you're not going to get to have them stop and take a good look at them, or they're puddling in damp sand. But the size is really important on this one. It's about the size of a question mark. It is, as I say, noticeably bigger than a cabbage white. Particularly for people in northern Illinois, if you think you are seeing a cloudless sulfur, specifically ask yourself, is this the next butterflies that we will be looking at, a common or an orange sulfur rather than a cloudless? So common and orange sulfurs have a number of things about them that are challenging. In most of Illinois, this is the commonest sulfur species. That appears not to be true as you get into the far southern part of the state. But for most of our more northern monitors, this is what you are going to see most of that's a sulfur. Why is it hard? Well, for one thing, it hybridizes a lot. The common sulfurs are mostly yellow. The orange sulfurs are mostly orange, but you can get a lot of stuff that's kind of in between in some of these hybrids. There are white females in both of these, and the white females can be extremely difficult to tell apart. The black border around the edge, I think I've got that in the next slide. That can be really hard to see in flight. And I think this may also contribute to misidentification as cloudless sulfurs. You see it fly by, and often, even if you're getting a good look at it, they beat their wings really fast. It can be really hard to pick up that dark border sometimes. And finally, there is confusion added to the picture because there are multiple common names for both of these species. The common sulfur is often also called the clouded sulfur, which again with the word cloud in it adds even more confusion relative to the cloudless sulfur. And the orange sulfur is also called the alfalfa butterfly. So how do we deal with these challenges? The main way we deal with the challenges of the two species being so difficult to pull apart is that we don't. We simply lump these two species here in the Illinois Butterfly Monitoring Network. They are here largely because of agriculture. They really like disturbed habitat. They are ecological equivalents in Illinois. And so it doesn't get you a lot of information to start really agonizing over separating the two out. In the menu, in pollard base, what you want to be looking for is common/orange sulfur. And I think that may also be the problem if you're starting to type and are looking for the clouded sulfur. You won't find it on our menu, but you will find the cloudless sulfur in our menu. The answer you want when you are recording one of these is common/orange sulfur. Again, size is a very important factor. These are almost the exact size as a cabbage white. And so, that's a good familiar species that's a nice size landmark that you can use when you're out in the field trying to identify these. Again, do not make the call that it's cloudless just because it's very yellow. Use more characteristics than that. Common and orange sulfurs tend to fly lower. You tend to see them waist high or lower. They land a lot more. They may not land for very long, but they're avid nectar feeders, and you will see them on flowers a lot. But even if you see them flying, you don't tend to see them up as high as you do the cloudless sulfurs. And this is just some pictures to give you an idea of the range of appearance that these can have. That's a white female. That's another white female. And that's it.
All right. Now, I know on the agenda slide, we had another break right now, but I think in the interest of time, I'm going to power through if everyone's all right with that. So let me get my slide showing, and I hope everyone is ready to switch gears a little bit into a completely different taxa. I thought it would be fun to talk a little bit about cicadas and do a look back at the emergence convergence that we experienced here in Illinois, throughout Illinois, in 2024. Oh, okay, let's hide this presenter view. There we go. Now can this go away? Okay. So I don't know if y'all remember this, but it was all over the news for a while. It kept me very busy. I was doing the circuit, bringing an interpretive box of cicadas that we have here in our Nature Museum Beecher Lab that we can check out for interpretive programs. And it was just really fun. I really liked having the opportunity to talk, to folks about insects and see the range of reactions that we can get. It was also bringing live cicadas when talking to the media at the right time of year. So all this news was specifically about a group of cicadas called the periodical cicadas. These have such long lifespans for an insect. They live for either 13 years or 17 years. Now it's the more southernly distributed cohorts that live for 13 years. It's the more northerly distributed periodical cicadas that live for 17 years. And they're going to be active kind of early in the season as far as cicadas go. So late May through June. It's really not that long of a period, although folks who live in the thick of it may have felt much longer. Now, they're called periodical cicadas because they have these periodic synchronized emergences. What periodic means is that it happens regularly. in certain time intervals. So again, every 13 years or 17 years, and they're synchronized. So they all really kind of come out at the same time. And there are multiple populations of these. So it's not all periodical cicadas across the entire country come out at the same time. There are distinct different populations that are grouped together called broods. So the different populations that emerge at the same time, kind of in the similar area or contiguous area, those are going to be defined as a distinct brood. And here's a map showing, some of the different the different broods that are in Illinois. And then here's a map from the U.S. Forest Services showing all those different broods across the U.S. So this is really an American phenomenon. There isn't any periodical mass emergence of cicadas that occurs anywhere else in the world to the degree that it does here. There are some cicadas that will have mass emergences, like say every 7 or 9 years or so, but not quite as synchronized as ours. And all the different colors here, that is a different brood. So in Illinois, we have 5 different broods of periodical cicadas, which is tied for the most of any state. We have 3 broods of the 17-year cicadas and 2 broods of the 13-year cicadas. So those 17-year cicadas are going to be in the northern half of the state. Those 13-year cicadas, think southernish half of the state. And what was so special about last year was that we had a co-emergence with a 13-year brood and a 17-year brood at the same time. And this only occurs every 221 years. Now, there's nothing, it's magical, but there's nothing that's like super coincidental about that. This is just 13 times 17. It's just how often those cycles happen to line up. But the last time this happened, between the 2 broods that we have in Illinois that had co-emerged last year was in 1803. So that was, just for frame of reference, the year of the Louisiana Purchase. Then we had 2024, a couple years ago, and the next one won't be until 2245. Who knows what the world's going to look like for us as people in more than 200 years. Now, the next co-emergence, though, it's not going to take quite that long. The next co-emergence we have is in 2037. That is brood 19 and brood 9, except those are not adjacent. Those are 2 broods that are not kind of really close to each other. The next adjacent co-emergence we have, that's going to be in 2076. So we got a while for that one. And just what was so cool about this phenomenon is that it provided so many opportunities to do outreach and, just talk about insects into the public. We did a lot. I did the circuit. I got some more photos coming out later, but it was really just fun to see entomology departments, science museums, cultural institute, like all sorts of cultural institutions, schools all come together and talk about cicadas. There were even some really interesting pop science books that came out. A really good one that has sort of all the information you would ever want to know about these 2 broods is A Tale of 2 Broods by Gene Kritsky. Highly recommend it. And for wider audiences, there's even a graphic novel, Cicada Apocalypse, which was sort of a look at what this co-emergence looks like from the perspective of a squirrel, like who doesn't know what's happening. But there is some really good information out there on cicadas. They are pretty well studied as far as insects go. I just want to do a shout out to the cicada page by the University of Connecticut. Their premier cicada researchers are here or there. Check out this website. This is where I would go for the most sort of up-to-date scientific information on our periodical cicadas. But if you want to have some more fun, there are sites like Cicada Mania that have sort of a little bit more broader focus. They have a news feed where around the world, any fun cicada events that are happening or other brood emergences, those will be listed here. So that's fun to check out periodically and see what's going on. They also have information on all the different species of cicadas you can find around the world with their calls. So you can check out this website and listen to what all the different cicadas sound like. And if you go to that website, you can find information on all the cicadas found in Illinois. believe it or not, we have 22 different species of cicadas here in Illinois. The ones that probably most of us are familiar with or have heard sort of in the evening hours are this group that belong to the dog days, or they're considered the dog day cicadas. And there are multiple species and 2 genera within that group, the Neotibicin and the Megatibicin. So Neotibicin, those are really the dog day cicadas. Megatibicin, they're big dog day cicadas. Although there's one here, Megatibicin dorsatus, that this is the grand prairie cicada. This is one that you will find on high quality grasslands. Yeah, like good remnant habitats. Those are going to call more during the day. Their call is said to sound like a spoon on a washboard. Really gorgeous. These cicadas kind of hang out lower in the foliage. They're not going to be so up high. So if you do go to a site that has them, they're not terribly difficult to observe. Belmont Prairie, shout out. So a little bit more information on these dog day cicadas. They also get this informal name called annual cicadas. And this can refer to more than just the dog days, but generally when people say annual cicada, they're referring to the dog day cicadas. And they get this name because they have unsynchronized emergences or asynchronous emergences. So some will be coming out from underground every year. So you don't have to wait to hear them every summer. You're going to hear them. They have pretty long lifespans as far as an insect goes. That usually ranges from about 4 to 7 years underground, above ground, just a couple months. They're going to be active later in the season than the periodical cicadas. So July through August, or at least until it gets too cold for them to function. There's usually some hanging on pretty late in the season. And these look a little bit different. Those periodical cicadas are small for a cicada, but large for an insect. They're primarily black and orange. These annual cicadas, they're going to be more like greenish, brownish, and be pretty large. Now, individually, since they're large, these cicadas are louder than the periodical cicadas, but there are so many periodical cicadas where they emerge altogether, that sound is deafening and cumulatively louder. So this is a chart showing some of the phenology of some of the cicadas of Illinois. So those local periodical cicadas, remember, those came out in May and then through June, and they were kind of done. So they're not really out at the same time as most of our dog day cicadas. Now, down here, these 4, these are some other species of cicada that you're going to find more on high quality habitats, not in the same place where you're going to find the dog days. Cicadatana, a tiny cicada about the size of like the tip of my finger. They have such a high pitched shrill that I need a test to see if I can still hear them. I'm at about the age where most people stop being able to hear them out in the field. So it's helpful if you can bring an undergrad with you. Down here, Neotibicin pruinosis, that is one of the dog day cicadas that is most well known. I'm going to do an embarrassing impression here, but it's the one that kind of sounds like a leaf blower to me. They kind of go like, and so on. Okay. So back to periodical cicadas. There are 7 different species of these. In the 17-year broods, there are 3 different species. In the 13-year broods, there are 4 different species. Now, there are 3 different species groups total. So if you look at the scientific names, what you want to pay attention to is the end of the name here. You have your decim, and then for each 17-year cicada, there are the 13-year complements. So they're pretty similar to each other. So we have the 17-year decim, septendecim, that just means 17. And then there's the 13-year versions of that. There's actually 2 decims, neotradecim and tradecim. So there's the new 13-year and the 13-year. We have the Cassinis, and then we have the Deculas. And Illinois has all 7 species of periodical cicadas that exist. Now, they are really hard to tell apart physically just by looking at them because they all are going to be, orange and black cicadas emerging at the same time together. So if you do want to tell them apart, here's how. First, you want to look at the side of their head, sort of like this neck cheek area, and see if it is orange. If it is, you have a decim. Boom. If it's not, it will be black, And the next step is to look at orange striping on the abdomen. So the Cassini and the Decula groups, they should be black on the neck here. And then the Decula will have orange stripes and the Cassini will just be black without orange stripes. Although I wish it were this easy, it actually is a little bit trickier. The male Cassini, some of them do have some faint stripes. So they can play trickster and it can be really difficult to tell them apart. But if you look carefully when they do have stripes, they're not all contiguous. They don't all make it, they don't all quite attach to each other from both sides. Let me go back here. The Decula that's going to be a bit more prominent band that goes all the way across the abdomen. But sometimes you really can't tell in the field. You have to either set them down and wait to hear what sound they make, or bring them back to the lab and sequence them or something. There are more than 3,300 species of cicadas worldwide. And what's so cool is that almost all of them have a unique song. And by listening to their call, that's actually the best way to tell a lot of the species apart. So I'm going to play you the calls for the different members we have of the 17-year cicadas. So first up, we have Magis cicada septendecim. This is also known as the pharaoh cicada because it's said that their call kind of sounds like they're going pharaoh, pharaoh. And now when there's a lot of these all together, you kind of hear them just make sort of a low hum in the background. So it'll be like the whoo droning in the background. Those are the decims, and here's what it sounds like. Oh, hold on. There is an option here. I have to check when I share the screen. Share sound. Okay. Again. All right, I was testing this earlier and got it to work, and it decided it doesn't want to today. So I'm going to be doing embarrassing impressions for the rest of this. So this one went pharaoh, pharaoh. Now the Cassinis, these are much smaller cicadas. They're going to have a little bit higher pitched, and they're going to kind of do some clicks and trills. So they're going to kind of sound like click, 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 and these are really loud and oftentimes can kind of overpower the other cicadas. As you get closer to like a big group of them, you're going to hear a lot of those. And then last, again, the Decula. These were less common in the 17-year brood that we have here in Illinois. These are just going to kind of make a click. They're going to be like click, 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 click. And oftentimes it's hard to hear that in the mass of other cicadas. And then the 13-year cicadas, they're going to sound very similar. Now, how do they make these sounds? They use this organ that's right here behind the wings on their thorax called a timbal. And the timbal is a stiff membrane with all these ridges and folds that has muscle attachments all around the edges of it. And what the cicada does is, pull on those muscles to deform that membrane and then relax it. And as it sort of gets pulled and snaps back into shape, it makes a sound. Think of like when you push the top of a mason jar lid down and you hear that little like doot, 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 doot. Imagine doing that really fast. Or if you have a thing of sheet metal that you're rattling. And then behind that timbal, they have these air cavities called air sacs. So cicadas have these large abdomens because they are largely filled with air, at least portions of them. If you pick one up, they might be a bit lighter than you would expect something that big to be. And those are sound chambers that help the sound resonate and then releases out, amplifying the sound, much like the chambers of a guitar, violin, cello, and so forth. So they're living instruments. All right. So what actually, happened during this emergence? What are they doing? If we had audio, you would hear all the calls together singing in a symphony. They're having a party. They're doing their thing. They're eating, they're mating, laying their eggs, then they kind of die and they're done. So here's just a clip of them all hanging out together, kind of calling, and they'll form chorus centers where a lot of males will congregate and sing together, and the more that are singing together, the louder that sound is, the further it carries, and then from further away, they can draw in the female cicadas. All right, we're on low battery, even though I'm plugged in. Power strip is now on. Okay, and now what was happening during the emergences, talking about public outreach, I want to give a shout out to DuPage Forest Preserves. They put out some incredible videos on the life cycle of 17-year cicadas. Check this out. I can't really describe what's going on here, but it is great. And it was so fun. It was even featured on the Colbert Show on TV here. So that was an awesome moment. Okay, so what's going on is around mid-May, soil temperatures get warm enough. That is the cue for the nymph cicadas underground to crawl up and start the emergence process. And you might see their little emergence holes show up before emergence starts because they will come up to the surface to kind of check things out, check out conditions, and then they may go back under. There was some news where some folks, one institution, saw the emergence holes. Oh, all the cicadas are on the surface. They announced, hey, the emergence is happening. Everybody, news started contacting, made everybody jump. And it was like, no, they're just checking it out like a week or two, almost, but not yet. But it's when that soil temperature 6 inches below the surface reaches 64 degrees Fahrenheit. That's kind of on average when they come out. Oftentimes that's achieved when there's a rain that sort of infiltrates the soil. And most of them are going to come out sort of late afternoon into the evening and overnight. So when they emerge from the ground, what they're doing is they're crawling around and looking for a vertical surface to climb up and latch onto. You may notice these big expanded front claws here. They're both fossorial, meaning they help them dig through the soil. So they're like little shovels, but they're also raptorial, which are really good for grabbing. So once they climb up to a surface, they can really latch into that surface and hang there nice and sturdy. By the way, a lot of these images came from Brood 10 that I visited in Indiana. I believe that was in 2021. So I was prepared for the emergence convergence of 2024. And when that happened, I took one back to take inside so I could do a time lapse of the eclosion process. So what eclosion is that final molt of an insect's life stage when it becomes an adult. So that nymph will crawl up to a surface, grab on tight, and begin the process. And what you're going to see here is the back splits open here, and then the adult, as it emerges, it's pumping its hemolymph, which is the fluid that fills an insect's body cavity, kind of like our blood. It's pushing that through its body to kind of bulge out here. So here's its back coming out. Its head is up here, and it's going to get the wings out pretty quick in a moment here. You see here are the wings here. There's an eye. And it's going to kind of come out backwards, and it's going to do some gymnastics and hang. So, front legs are coming out now. And you can see those motions of it pushing the hemolymph through its body. They're kind of coordinated movements that are hormonally controlled, these behaviors. So here you go. It's pushing out. Now it's going to hang back, and that's going to release the wings. And now it has to pull all of its legs out. And as it stretches a little more, you're going to see some of these little strings. And what those little strings are is the lining of their trachea. So the way insects breathe are through little holes along the side of their abdomen. And there's a series of tubes called trachea that are lined with cuticle that they have to shed those too every time they shed their skin. So imagine as you grow up, if we had to shed the lining of our lungs all the time, that would suck. Okay, so after a moment of time, then they reach back up and climb onto the surface. They might climb around, they might hang out. And now what they need to do is pump that hemolymph through the veins of their wings to expand them. So you saw how they were really, kind of small at first. And now it's going to slowly start expanding those wings. This is a process that you can see butterflies do if you visit the Butterfly Haven here at the Nature Museum. And now they're expanding here. And then once they're done expanding, you're going to notice the wings go from flat to more tent-like covering their abdomens. I'll just fast forward a little bit so that we can get to that. And you'll notice here that this is pretty pale in color. This is at a stage, it's called teneral. They're nice and soft. These are edible insects. If you were to eat them, this is when you want to collect them so that you don't have to like deshell them or anything. So there you go. The wings were done. They kind of snapped into place. And then it needs to hang out for a few more hours for the chitin in their exoskeletons to form these cross linkages, which is what makes them rigid. And the cuticle is going to tan at that point, and then that's when the color kind of develops. So there you go. That's that process that happens to happen at night. And they leave behind these exuvia. These are the shed skins of the cicadas. And during a periodical cicada emergence, these really appear kind of everywhere. You might find them from some of the dog day cicadas from time to time on trees and fences. What's kind of fun is you can actually identify cicadas kind of decently, depending on the species, from the exuvia. Like you can look at the size and shape of the nymphs. You can extract DNA from them. So there's a lot of use we can make by collecting these. I also like to collect them because they're just kind of fun to, put on my shoulder, hitch a ride, or stick on a friend, or put on my cubicle wall as my little buddy. But now, not every cicada that tries to emerge is going to make it, especially in these really dense periodical cicada emergences. Sometimes they crawl on top of each other and they interrupt that process. When they are soft, when they're teneral, they are very delicate. Mishandling them can prevent them from being able to expand and stiffen their wings in the proper shape that they need to fly. Also, if they can't find a vertical surface, say like all the trees had been cleared within the past 17 years and they have nothing to climb up. Here are a bunch that congregated on this one post kind of in a lawn. They might not all make it. Okay, so if they do make it though, now what are they doing? Well, they got to eat. Things that most animals do to live. The way they eat is with these piercing, sucking mouth parts. So cicadas are in the order of insects called Hemiptera. Those are the true bugs, and the defining feature of those is this sort of piercing, sucking, needle-like mouth part. There's two parts. This can look pretty intimidating, but thankfully, cicadas are vegan. They don't bite. Some species, on occasion, if they're hungry, they might want to explore. They might try to poke, but they're not really going to pierce all the way through. What they are looking for is the fluid that is found in the xylem of plants. And this is a very nutrient poor food resource. So they have to process a lot of it to accumulate the nutrients that they need. And as nymphs, they have the same kind of mouth parts. They're feeding on the same kind of fluid, except from the roots below ground. So because of this, they excrete a lot of water. If you go online, you might see some cool videos of cicadas peeing off of trees with great force. They kind of spray it pretty far. It's entertaining. Okay. Now, the big point of the party, though, is courtship. A big benefit of having these synchronized emergences is after spending 17 years underground, when they come up, it's kind of guaranteed that mates will be available. They're not going to come up above ground and nothing's there. So I have a little clip here of some courtship in action. Now we can't hear it, but there are males here and here calling. And if a female is receptive to that call, they may approach or they're going to do a little wing flick. And when they flick that wings, that percussive sound attracts the males to approach, and then they may initiate mating. This can be co-opted by researchers. Sometimes cicadas can be hard to collect because they'll spend time high up in trees. Well, if you happen to find a male that's high up, if you get the technique right and you can snap, or what some folks will do is bring out a light switch and flick the light switch, the male cicadas might get curious and come down closer to you so you can at least reach them with the net and catch them. And this is sort of how it works across cicadas, so not just periodical cicadas. So you can try this with your dog day cicadas. So when they mate here, it's end to end. Insects, really do kind of have lock and key genitalia that fit together. And they may stay this way for a few hours. And then after they've mated, then the female cicada will then go and lay her eggs. So she has this structure on her abdomen called an ovipositor, and she uses that to stab into twigs and branches, and that's how she can deposit her eggs. So she's pumping her abdomen, and eggs are flowing through there into that branch. So this is how these periodical cicadas can cause some plant damage. Now, their feeding really doesn't cause significant damage, but the oviposition can. Now, for most healthy large trees, there's visible damage, you'll see it, but it doesn't actually impact the overall health of a tree. But what it can have an impact on are very young trees or sick trees. So Morton Arboretum, also jumping on board with the outreach, really got the word out on how to protect your plants from cicadas. You can use netting, and that can help exclude them. Although if you are really in like the epicenter of where they're emerging, they're going to be crawling all over the netting. Some of them might be able to, fit through that netting. It's not always going to be successful, but it really does help. It was kind of funny. I did see a lot of trees netted like within the city proper, because the word had gone out so much, but there weren't that many cicadas here in the city proper. A little more on that later. But what this oviposition does is it does create scars. You can see those visible on the tree here. And then that prevents nutrients from making it all the way to the tip of the branches, so the tips of the branches can die back. Oh yeah, here's a video of oviposition here, and it's forming those scars. So, but what these scars are physical signs of the egg nests that the cicadas laid. Each individual cicada may lay up to 20 eggs per nest, but they're going to lay multiple nests. So you can get up to like 600 eggs per cicada. Now, when you have like millions and billions of cicadas emerging at the same time, that's a lot of eggs. So those eggs will hang out in there for a few weeks. Eventually, the nymphs will hatch out of those eggs, and then they fall to the ground and crawl underground. Okay. The tip of that twig may die, the leaves will turn brown, and it'll hang down, and that's called flagging. That's sort of a characteristic damage that cicadas can cause. But generally, control isn't needed to prevent this. Again, you may want to protect young trees. If you had plans to plant an orchard, you might not want to do it the year you have two broods of cicadas emerging at the same time. So just maybe delay that a year later in the season if they're the kinds of trees that can handle that. But just of note, I'm not aware of any pesticides that are specifically labeled for use on cicadas, and that's not really an appropriate way to control periodical cicadas. You just kind of wait for them to go. They're only going to be around for a couple months. Whenever applying pesticides, it is really important to always follow the label, the directions on the label exactly. Like that is a federal government contract. By following those, we can help limit our non-target impacts that are causing declines on things like butterflies, as we've seen. So they can cause a little bit of damage to plants, some severe damage to unfortunate plants that are in the wrong place at the wrong time. But can they harm us? Answer is no, thankfully. Caveat being, if you have an allergy to insects, maybe don't eat them. But these are just incredible to observe because they really have weak defensive behaviors. They're not likely to fly away from you very far when you approach them. In fact, if you walk up to a group of them to try and take photos, they very well may just fly at you, bump into you, land on you. You can just walk up, pick them up. They're really fun. Their survival strategy is kind of safety in numbers. If enough emerge at the same time, just whatever can eat them can go ahead and eat them. And then there's still enough remaining to survive and keep the populations going. This is called predator satiation. So all the predators are just too full to eat them all. Now that only works though when you have them emerging in high enough numbers. In areas where they still exist, but maybe not high density, they do get eaten pretty quickly as they emerge, so we might not see them, even if they're there. So a big question in the lead up is like, where are they? Are they going to be in my neighborhood? Are they going to be all over the city? This was my first time being here for these 17-year cicadas, so I wasn't 100% sure, but what we do know is that, or what we did know before, is that they are pretty patchy in distribution. So you're going to have some areas where there's a ton. You're going to have some areas where they're pretty sparse or they're just not there at all. But where they were denser, those are in forested areas that have undisturbed soils, or at least not frequently disturbed soils. And then the experience of the 17-year and 13-year broods, it's pretty similar. The species look the same, they sound the same. So as you travel like north and south through Illinois, you would hear all the cicadas calling at times. They might fade at times. You'll hear them again when you get into another epicenter, but you won't necessarily know when you've switched from where the 17 17-year broods are to where the 13-year broods are. It's kind of just like they're everywhere. Now, to try and determine where they are, this has been a historical effort. It's been known for a long time that these cicadas, like that this phenomenon occurs in the US. And one of the first systematic efforts at trying to map these was done by Charles Marlett here in the early 1900s. So we have a map here from 1907 ranging to 1922. He was the first one to sort of group these periodical cicada emergences into broods. And so this is where those Roman numerals came from for the different broods. He originally described 30 broods. So that's why the numbers are higher than the total number of broods we have. It's since been narrowed down to 15. With further analysis, we've figured out that what was split up as separate broods, some of those are really just one brood, but there are also a couple that have gone extinct since then. But do you think in 1907 that Charles Marlett could travel across the entire US or Eastern US US at the time to document this? He couldn't. So from the beginning, this relied on participatory science. Marlett did a lot of correspondence with farmers, people across the range, and had them report back on where they saw and heard cicadas and what information they had. And that's how he was able to construct these maps. So this really was, a community science project before that was known what it was. And these maps have been refined over the years. So at the Illinois Natural History Survey, we have the standard maps from the 70s. So these were some of the maps I've shown earlier. Here in the middle, we have others from Cooley and whatnot. So these they've developed from their own research where they're driving around with recorders on their car, picking up different cicada sounds. And they're also incorporating data from a community science app they developed called Cicada Safari. So folks can go on, submit their sightings, and those get incorporated into their data sets there. And also, like iNaturalist accepts observations for periodical cicadas. These are going to be a bit biased towards population centers because there are more people submitting data there. But still, it helps give us an idea of where the cicadas are. Now, a big question with this emergence convergence was, do the two broods of periodical cicadas overlap? Do you get 17-year cicadas mating with 13-year cicadas? Well, previous analysis showed that they do get pretty close, but it doesn't seem like they actually really overlap, at least not to a significant degree. There are areas where they come within about 5 kilometers of each other, sort of near Springfield and a little bit up near here. But it's found that these periodical cicadas, they rarely fly more than 50 meters from where they emerge. So they kind of stay put. They may move a little bit over time, but they might not make that 5-kilometer journey. And at the time, it's hard to tell when you're in the thick of it because the 13-year species look just like the 17-year species, but I have not seen anything coming out showing that there was, in fact, significant overlap during this coemergence. Okay, but I got asked a lot, where were they going to be in Chicago? Everyone was preparing for this. And then a lot of folks in the city proper were like, where are they? This was no big deal. This was overblown. And then everybody in the suburbs was like, what are you talking about? They're everywhere. We can't escape them. Well, there were very few in the city center, but they did exist. There were some that we could hear, but we have disturbed soils and constant development. So that first couple feet, if you excavate that soil, you're going to excavate most of the cicadas with it. So, and also when you don't have high enough numbers, again, most of them are going to be eaten before we can observe them. But out west, out north, a little bit out south, especially in the forest preserves, they did occur in great numbers. And there was the Singing Insects Monitoring Program put out a Magic Cicada Sound Map project to help document which forest preserves they were really occurring in. And so if you go on iNaturalist, you can look at this and hear the different call recordings that folks took and submitted online. And then there were some really fun findings out of St. Louis. There was a lab that put out a cicada mapping project as well. So through community science, what they found and were surprised by was that there was a vacant lot here and sort of like in a big urban area of the city that had high numbers of cicadas. Now, that lot used to have housing developments, which is what is pictured here. But about 50 years ago, those were torn down. And then for that 50-year period, that land was left pretty vacant and undisturbed. Now, in older parts of the city that have existed for a long time that have really old trees, they thought maybe there will be more cicadas here, but it turned out to be the opposite. Those older parts of town have some continuous development, over time each year. So that really keeps the cicada populations down. That land untouched for about 50 years, cicadas moved into there, populations built up, and then they were actually dispersing out from there into other parks around the city. So they have a great example here of how we can do conservation even for insects that have really long lifespans here. And they would not have been able to get enough data to do this if it wasn't for community science. Now, we get the question a lot also, like, why do we care? Other than they're here, what's their importance? I mean, well, first I'll say they're insects. We should just care about all species, but there are some things that are special to these periodical cicadas. One is they have an interesting role in nutrient cycling because they're feeding on that xylem that is nutrient poor. They're, tapping into this food source that doesn't harm their hosts very much. I kind of think of it as they're like taking a little bit of a tax, but not enough to really hurt the pocketbook. And then when they die, next slide, you'll see it, then they all kind of return those nutrients back to the soil at the same time as a nutrient pulse. So it's kind of like everyone's getting their tax return at the same time of year. They also utilize gut endosymbionts inside their bodies to process these nutrients that they're getting from plants. And that's an entire ecosystem there. There's biodiversity there. If the cicadas die, so do their endosymbionts that maybe can only live in cicadas. Now, probably their biggest significance is that they're a big food source. Again, like everything that can eat them they'll get satiated and well fed. There was a really fun study that looked at the gut contents of copperhead snakes from southern Illinois that were, in stored collections. So they're like in jars of fluid. They could cut them open, find out what bugs were inside, and some of them had eaten quite a few cicadas. And they were able to document that occurred during a big drought year. So if there's an emergence that corresponds with the big drought, this could help, alleviate those effects of the drought for certain animals. Also, again, I mentioned they're hosts to different microbes as well. Like there's fungi that specialize on cicadas. There's Massospora. It's a fungus that can fill up their body cavities, and then as they fly around, the tip of their abdomen may fall off, and spores will get distributed throughout the environment. But if they don't have the cicadas to reproduce in, that fungus will also be gone. Those fungus-infected cicadas are sometimes dubbed flying salt shakers of death. And then also just cicadas, they really add to the sound of summer that those dog day cicadas really kind of evoke, a lot of emotion and memory for me, just like end of the summer, back to school time. Cicada Summer is a band that just, really evokes that nostalgia for me. And also cicadas are important for art, culture, ecotourism. Again, this is an American phenomenon. People traveled from all over the world to come here just to see this. And here's an example of when they all die and you get that nutrient pulse. So for the couple weeks after the mass emergence, it can get a bit stinky, but over time that does wear away. And then, all sorts of events. I toured around, showed the cicadas, tried to get the word out on why they're cool. The Best Bower Dunn Museum of Lake County put out a really cool exhibit on periodical cicadas. That was really great to see. Had a lot of, interesting information, beautiful art, historical books here. The Insect Asylum did this project, the Cicada Parada, where they made hundreds, I think even thousands of these plaster cicadas that artists adopted and painted and were distributed all over the city. They gave me one in exchange for giving a talk for them. So I painted that one to look like a realistic cicada. So it's nice to have that as well. So this was just kind of like a unifying biological phenomenon. Never before have I seen groups from all walks of life like kind of come together to talk about this. If you go on Facebook, there were so many groups. There was, the Illinois Cicada Watch website for up to date, information on what's going on with this emergence. Local historical societies were pouring through their archives and looking for evidence of past emergences or writings for that and sharing that information. It was really cool. And for a period here, we had just a public that was really educated on kind of high level aspects of insect biology that we don't usually see, like the concept of different broods, periodicity, incomplete metamorphosis versus complete metamorphosis, the fact that there are 7 species of these things that look the same, like that's not something I knew until I specifically went to study them in college. Or ecological concepts like that predator satiation. And just all this interest is adding to, and the community science is adding to our ever-growing understanding of a pretty difficult taxa study. Like if you're interested in studying that northern brood of 17-year cicadas here, you only get one chance to do it every 17 years. So folks who study periodical cicadas, they travel around, they'll study multiple broods, but to study one brood, like 17 years, that can be a career. um, and just something else that I think is really cool is they have a pretty unique life history as far as insects go. This longer time scale, it gets us to consider our place in nature and the natural world on a different time scale than we usually do. I mean, with the Illinois Butterfly Monitoring Network, we're also getting there since we now have decades of data. And so this is really getting us to think about how land use change, how our relationship to natural areas changes over the course of decades, not just from year to year. And also not just on the geological time scale, like tens of thousands, hundreds of thousands or more years. It's really that in between, decades, hundreds of years. Okay, so thank you. Thanks for letting me geek out about cicadas here at our butterfly meeting. Yep. Oh yeah, so the question was from tenorol to full color, how long does that take? Just hours? And yeah, I would say, yeah, just hours. Hmm. Yeah, exactly. Exactly like dragonflies. Oh yeah. So the question is, they lay their eggs in trees. So does that mean there's a different species that's coming out of the ground? To connect that part of the life cycle, what happens is the nymphs will hatch out in the trees and then fall to the ground when they're really, really tiny, and then they dig underground and they'll spend the rest of their life there, or must. They stay underground with the rain. Yeah, they can. Oh yeah, when it's emergence time, the rain doesn't flood them out, but it's kind of a cue. Like it helps warm the soil and it lets them know like, oh, it might be a good time, and it'll just encourage them to come out. Yeah. Oh yeah. Yes. Yeah, so there are some drawbacks, of course, to every biological phenomena. So we had a question bringing up oak itch mites. That is a thing. So those egg nests, there are some mites that like to feed on cicada eggs. So when there is this ample food source, that means those mite populations can boom. So these mites that feed on those eggs can fall out of the trees if you hang out under them. And some people are more sensitive to them than others. The mites can bite and they give itchy bumps like chigger bites. Those are also a kind of mite. Yeah, you can get a rash. It can be really uncomfortable. It's not long-term harmful, but it is annoying. It's really annoying. Yeah. Although some people do have stronger reactions and it can be painful for them. Yeah. Did you have another? Yeah. So we had a question is when they crawl underground and as nymphs, they're feeding on roots. Will they travel from plant to plant? Do they kind of just hang out on that tree? And the answer is, yeah, they do travel around from plant to plant. They're not very, they're not picky about what they use as a host to feed from. So they'll kind of tap into any root and not just woody plants. Yeah, herbaceous plants as well. So they'll feed on grasses and forbs and whatnot. And when they emerge from the ground, if there are tall enough grasses, they can crawl up grasses as well to hang off of. Yeah. Any hands in the room? Let me go to the chat here. I see some stuff pouring in. Dude. Ooh, Phyllis Schulte was able to find a blue-eyed cicada during the 2024 emergence. That's really fun. I know it's kind of like a 1 in a million. When you have hundreds of millions, you're going to find some, but it's still a very rare occurrence. That's really cool. I think we added two to our collections as well for the Academy that were found and submitted to us. So that was really fun. I know the Field Museum was trying to collect some as well so that they could try and study what the mutation is that causes that blue eye pigment. Oh, okay. The sound did play through Zoom. So if anyone who sticks around can hear it. So that must have been annoying to hear my impressions over that sound. Just wasn't playing in the room here. Oh, someone made cicada cookies as well, cicada stir fry. That's awesome. You know what? I didn't actually eat any of the cicadas. I've like made chocolate chirp cookies out of crickets, but I hadn't tried it with cicadas. There was a cooking class I wanted to go to. They were gonna do some cicada ice cream, I think, and maybe a baked good, but I was giving a talk that day. I couldn't go. Okay, supposedly St. Louis experienced more lightning bugs than usual during the emergence because there were so many cicadas for birds to prey on and birds were eating them instead. Yeah, that's true. So that when everything has some big juicy meal to eat, they're going to eat that and other critters might benefit from that slightly. There were actually some drawbacks for birds. Wildlife rehabilitation centers, we're seeing an uptick of birds with eye issues that they linked back to a vitamin A deficiency. So it turns out this cicadas as a food source aren't very rich in vitamin A, and they're pretty good meals. So it was mainly like blue jays and something else that I didn't care about is starlings. It was starlings and blue jays that were affected. I felt bad for the blue jays. Um, but yeah, there are trade-offs with everything in nature. Let's see what else we got. Oh, that was it. Do we have any more questions? Okay. Well, with that, I just want to say again, thanks everybody for attending so much. And we really appreciate it. Our spring training season is posted on the website. If you go to ibmn.org, we have our first introductory workshop next Wednesday on the 18th. Starting at, I can't remember if it's 6 or 6:30 p.m., but somewhere around there. Check out on the website, register if you want to get started with that. Doug, did you have any concluding remarks? All right.
And my concluding remark is we did not do this deliberately, but this year we happened to time the annual gathering of the Butterfly Monitoring Network to be on National Learn About Butterflies Day. So happy National Learn About Butterflies Day to all. Thank you so much for coming.